All right. So, yeah, liver panel. Like, oh no, the liver has problems. We're gonna do tests. What do the tests tell us? That's what we're talking about. And this is totally a memory technique when you do that. Anyway, yeah. So liver panel. We got a few things here, and I know you can't see it, so I'm gonna write over it in a better marker. So we have 3 groups of tests. First, we have the first group is asking kind of, are the liver cells alive? Are the liver cells okay? All right. And the 2 tests predominantly we have in here are AST, and there is a third one, but I'm going to talk about these 2 mostly, and ALT. AST is aspartate aminotransferase. ALT is alanine aminotransferase. And for AST, that way it doesn't look like the letter I over there. All right, for AST, if we have a picture of the cell up here, we have a mitochondria right here. And in the mitochondria, as you might remember from environmental or some other classes, the mitochondria is where we do detoxification. We have most of our CYP proteins and enzymes, CYP, P450, etc., the detox stuff. And one of those that's involved in detoxification, it's not a CYP, but it's involved in detoxification in the mitochondria, is aspartate aminotransferase, AST. So if we have really high AST levels, it's because the mitochondria is dead. You know, it's because the mitochondria has let its enzymes go out, and those enzymes have no other place in the cell, so they'll make their way to the blood, and so we have high AST levels. So that's going to be drugs and alcohol and anything that's toxic. That'll be high. So mitochondrial damage, and if the mitochondria die, then the whole cell will die, and that's necrosis. And next we have ALT. ALT is found in the cytosol, and that will be released just inflammation. And if the cell dies altogether, like necrosis, we see more AST because there's more of those than we see the ALT. The ALT will go up a little bit. That's not what we associate it with. And there we go. I will call that back in just a moment. So ALT is with inflammation. AST is with toxins and mitochondrial damage, and then because of mitochondrial damage, necrosis. All right, so these are the tests for are the liver cells okay?
I think with AST, like necrosis.
Necrosis. Oh, I see. AST, necrosis.
ALT, inflammation.
Cool.
So, I'm like.
All right, go ahead and do that. You can also throw an ALA into this one. Alanine. No, wait. ALA is something else. But ALA, if that one's high, then it means liver cell damage, kind of like ALT, but it could also mean bone damage. And you don't know which until you know if the liver's having issues. And it's like, okay, it's probably coming from the liver because the liver's already having issues. So that's kind of a side note. Liver, that's the first chunk. Next chunk are ALK-phos, or alkaline phosphatase, bilirubin, and GGT. GGT, all of these are, the first 2, ALK-phos and bilirubin, those are normal things to go out in the bile. It's not weird to see them at all. If we see them in the blood, that means that they can't go out in the bile. GGT also goes out in the bile, and it has some special notes. So right now, all of these, it's abnormal to see a lot of GGT, though. The other ones are normal in the bile. GGT is like, that shouldn't be high ever. But all 3 of those are supposed to go out in the bile if they exist. And if they can't go out in the bile, and they'll show up in the blood. So if we see them in the blood, it means the garbage chute is plugged. So the bile duct is plugged. That could be because of cirrhosis of the liver, really, really, really bad fatty liver, more likely cholestasis, or gallstone, or impingement of the common bile duct by pancreatic cancer growing and pushing on it. And so these guys are going to be post-hepatic issues type things. So these are the bile leaving, bile can't leave, rather. Okay, so those are alk-phos, bilirubin, and GGT. And I won't write them all out since I wasn't doing that while I was talking. The last thing, GGT has a special thing. So we've done group 1, we've done group 2. Now we're going to do a special disease state that uses some from both of these. So we're taking our knowledge here and applying it. If we see high GGT, and that is very high, and then we have high AST, and that's not quite as high, but still high. And then after that we have a little bit less, but still high ALT. GGT is the highest, followed by AST, followed by ALT. This pattern is very characteristic of alcohol liver inflammation. So
Alcohol hepatitis.
alcohol hepatitis. So with the GGT, this is called gamma glutamyl transferase. And it is seen when your glutathione levels, most of your glutathione is oxidized. And so it hasn't been recycled and it can't get rid of antioxidants anymore. So if you have a lot of alcohol, you've used up most of your glutathione, and so then your GGT levels go high as your glutathione levels go down. And so this is actually, there's a research article that I'll put into the description below that you can read about this. So you don't have to take my word for it because this wasn't mentioned in class. But GGT is a good marker for general oxidation stress. If you have high GGT, you probably have oxidation going on in your body. You don't have enough antioxidants. And your glutathione isn't in a functional state. And so with alcoholic hepatitis, we would expect GGT to be very high because you've used all the glutathione to try and take care of all the oxygen stress that comes from alcohol. So GGT is going to be high for two reasons. One, oxidation stress or toxin stress is what I'm going to go ahead and write. Toxin stress, oxidation stress. I'm not going to rewrite it. And number two, bile is slower to leave in alcoholic liver disease. Slower to leave. So it's got two reasons to be high, and that's why it's the highest one of all. This one was from the research article. This one was from Professor L. Okay. Next, AST. This one's going to be high because what does detoxification? Well, liver cells, yes. But what part of the liver cell? Mitochondria. And so if your mitochondria are the ones that are doing all the detoxification, they're going to take the most damage and they're going to die. So mitochondria die, and that leads to high AST. And then ALT, you're going to have liver inflammation from just having your liver being overloaded with alcohol and toxins. And so it's going to come from that inflammation. But it's not a targeted sort of thing. And besides, there's less ALT than AST total. And so we're popping the mitochondria open with this one, but this one's still definitely getting out too. You just won't see it as high in the blood as mitochondrial one. And so highest, because there's two reasons. Next, because the mitochondria have a bunch of it and they're targeted in the alcohol liver disease. And then ALT is next, just because you are damaging the liver via inflammation and stuff, and so it's going to get out there. So if you see this pattern, highest, next highest, all elevated, that is classic alcohol liver disease. And this is used reliably. And I'll link below again that study. So we've now used our knowledge of these first two chunks of liver tests. Now let's move on a little bit to remaining tests where we have total protein that I'm not going to talk about a lot because I don't have a lot to say about it. Next, albumin levels in the blood. And then PT, and I just call it PT7 because I like to remember it that way. If I always call it PT7, I'll always remember a certain thing. And then platelet count. Platelet count. Now all of these, the third category is liver synthesis. Can the liver cells make anything? Maybe they're not dying. Maybe the bile's getting out fine, but you're still not making albumin. Your prothrombin time is still extended, which is bad because you can't start the coagulation cascade. And your platelet count is still low, which is bad, so you can't clot because you don't have any platelets. That would mean maybe you have malnutrition, protein deficiency, so you can't build anything, even though your liver is fine. And so if these are high and these are low, then that could be going on. What if your liver cells are dying? Liver cells are dying. Would you expect liver synthesis to go down? Of course. So if these are high and these are all having issues, then it's probably because the liver cells are dying. You know? All right. Let's talk about them for a bit. Albumin, of course, that's the protein in the blood, and that'll hold water in the blood. If you lose albumin, you're going to have ascites, and you can eventually have some cranial edema and stuff like that as well, but especially ascites and generalized edema. Prothrombin time, and I put a 7 after there. That's because prothrombin time will be extended because the liver is not making coagulation factor, clotting factor 7, and that's tissue factor. That's the number 7, and it's found in the tissue and of the extrinsic pathway. So PT7, full name would be PTINR, and I throw 7 on there. To remember that that is the pathway that uses clotting factor 7. And so if your PT time takes longer to make a clot, then factor 7 is low. Okay? So albumin, PT7, next platelet count. That one, if you don't have platelets, can't make a clot. So your clotting time will take longer in that as well. Yeah, so those are the 3 kind of categories of breaking down what's going on with your liver. Let's talk about, yeah, so those are the tests. Now, we've had one disease example. Let's have a couple more disease examples and then we'll tie up. So, if your liver cells are being killed off, what's the most likely thing that's going on if AST and ALT are super high? And then what's the most likely, what's a possible thing if they're just a little high? Okay, we're going to talk about both of those. So, super high, all right, we're just going to go over here like this, and we're just going to super and little. So we're making a little chart here, and I honestly don't know if I'm going to put more than one thing. But super high is going to be hepatitis, and this is going to increase by like 10 times. 10 times. So this will be like, if the normal is like, the normal of ALT is roughly 50, AST is in the same ballpark, so 500. Would be like hepatitis, 500, 1,000, 2,000, that's hepatitis.
Yeah, I think it's going to be a lot more.
Hepatitis.
In the case he
For
said,
which one?
For ALT, he said, so the first case, he said 1650, and normal was 33. So it's like...
Oh yeah, it's usually
it's
higher.
really, really
I talked to him,
high,
he
so.
said it'll be at least 500, I talked to him after.
Least. Oh, okay.
And
for
then
hepatitis.
if it's not at least 500, don't even think about hepatitis, because it's like, it should be at least 10 times the normal. Otherwise, hepatitis will put it, oh, I can't do his accent, but hepatitis is going to have tons.
Mm-hmm.
But yeah,
Okay,
yeah,
so
the
500
cases
is the
totally
limit.
showed higher.
Okay.
So 10 times at least. Thanks for putting the at least in there since I wasn't clear.
Okay, problem.
Yeah, and then a little bit if you have like up to 100 or something like that, and these are very rough numbers for this one, and that's probably like fatty liver disease. That could be fatty liver disease, and so that's something going on. Something else, was there, yeah, hepatitis is the most likely for that. Moving on to bile. For bile, I'm going to go ahead and just do the whole pre, and then liver, and then post hepatic jaundice story. So we haven't mentioned jaundice yet, but we're talking about the liver, so of course it's going to need to come up. So if you have these guys, we're going to start with post because that's what ties in over here with alk-phos, bilirubin, and GGT. So if those guys are high, then we are going to have post-hepatic issues, which means the garbage chute is plugged, and so it can't get out, so it goes back up into the blood. So this is going to be bile duct plugged, and that examples of that are gallstone. These are the two most common. Hepatitis is the most common cause of high AST and ALT, and then for high alk-phos bilirubin, the highest are going to be gallstones and cancer of the head of the pancreas, because the pancreas is right there next to the bile duct. Pancreas. And I don't want to rewrite all your notes. This is supposed to be like a really quick understanding video to get your head around it, because I hope it's helpful. That's really what it's supposed to be. And, you know, yeah, so post. Now we're going to go into the little aside, like, what's this pre and post and liver thing going on? That is going back to if your BUN, your blood, urea, nitrogen, so you got urea in the blood, how much? If your blood urea nitrogen is out of normal limits, so it's high, and your creatinine is also high, then you're going to put those in a ratio. Well, if one of them at least is high, they don't both have to be high. But you're going to go ahead and put the nitrogen over the creatine, and then basically if that ratio is above 20, then that's going to be pre or post hepatic. You probably have had this before, most of you who are watching, so I'll zoom forward. But if it's above 20, then it's pre or post. And if it's post, these are the two most likely: gallstone and pancreatic cancer of the head of the pancreas. And then, however, if it's pre, just since we're talking about jaundice, that's going to be hemolytic anemia. So, and that would happen because red blood cells have a whole lot of hemoglobin in them. And they have a whole lot of nitrogen-containing compounds, and those are going to raise the nitrogen, which is on top here, BUN over creatinine. So if the BUN number is super high, then that's what's going to put it above 20, and so hemolytic anemia, that is going to be what we see when you got red blood cells dumping a bunch of nitrogen into the bloodstream. This is before any of the blood has reached the liver, and so that's why we call it pre-hepatic jaundice. Post is after it has reached the liver, but then it tries to go out the garbage chute, the bile duct, and it can't, and so it comes back. All right, enough on that. Liver, then, is what we look at with liver synthesis enzymes. Is there something wrong with the liver itself? All of these are actually liver. So if any of these are off, any of these three groups, then that means, let's try and use these three groups to figure out what's wrong. Whether it was, hepatitis, fatty liver, any of these are not liver, but, try and see what's going on with the liver in general. Anyway, so what I would review if you wanted to have a kind of a chunking thing, because I know some people appreciate the chunking. See if you can review what are the three chunks and what goes inside of them for liver tests. So you have liver cells okay, bile leaving okay, liver synthesis. What tests are in each of those, and what diseases match up with each test. And start with one chunk and move on. So for the first chunk, try and say where the enzymes are. For the second chunk, try and talk about the alcoholic liver disease. For the last chunk, try and talk about the coagulation cascade. do each of those 3. After that, you can then go to the disease table as your last chunk. Hopefully this has been helpful, and I wish you guys all the best. Cheers.